This is the second time I've been here. I spoke here 2 years ago to the Men's Breakfast Club about this book, The Stem Cell Dilemma, which was originally published in 2008, and the second edition, this edition, in 2011. So I'm going to read a brief passage from the book about really the embodiment of human creativity, Leonardo da Vinci, and the greatest creative act we know, and that is, of course, reproduction. Human reproduction had always fascinated him. In 1512, at the height of his powers, he drew the fetus in the womb. One of his most famous anatomical drawings, it depicted what he called the Great Mystery, a mystery in many ways more profound than the enigmatic smile on his Mona Lisa. With the fetus in the womb, the study of science, medicine, and human reproduction was brought to bear on that mystery. The navel is the gate from which our body is formed by means of the umbilical vein, he wrote. What Leonardo could not have imagined as he examined the umbilical cord attaching the fetus to the mother was that it is a treasure trove of stem cells, cells with regenerative powers that someday may eradicate any number of diseases. Stem cells that have already saved the lives of people with diseases of the blood and bone marrow, people like Molly Nash. And do remember Molly Nash. We'll come to her again. I had the great privilege of seeing Leonardo's drawings at the Royal Library in Windsor Castle some years ago. It was a terrific thrill. I'm going to pass this book around so you can see that the fetus in the womb is pictured there. So today, I would like to talk about and maybe answer three questions: why and how were babies, or basic embryos that became babies, genetically edited? Why did it happen in China? And what's next? Okay, first a glossary. Terms that are associated with this whole field that you have seen before, going back, actually some of the Brave New World goes back to Aldous Huxley's 1932 book, Brave New World. And in the years since, these terms have cropped up, and you'll be hearing me refer to some of them: genie out of the bottle, plain God, rogue science, Franken babies and Franken foods, which is a kind of a pseudoscience way of generating fear in people. Savior siblings we'll talk about, and on. Okay, first let's start with China rather than the edited babies. Why did it happen in China? A month after the news broke in late November that these genome-edited babies had been born in China, the New York Times published a story: U.S. officials warn health researchers China may be trying to steal your data. Basically, the NIH asked for a panel of experts to look into this. The threat to American intellectual property and research is not just in artificial intelligence, which you've probably heard about. It is also in the whole biomedical research enterprise. It's a very significant threat. In terms of China's interest in this area, if you look at the clinicaltrials.gov website, you'll find 24 clinical trials listed. Most of them just recruiting, the phase 1 clinical trials, just beginning. Of the 24, 12 are in China. 8 are in the U.S. In terms of scientific citations, China has just taken off. Now, they've surpassed the U.S. in terms of scientific citations, and of course the quality of their citations is not as high as ours, but they're making a real effort to lead in that area. They're very, very strong in primate studies. We don't do those the same way. We don't look at primate studies in this country the way we used to. We're kind of backing away from that. They have fewer qualms about doing research with primates, stem cells, cloning, and so on. This is an area that they're really surging in. In late November, a writer and editor for Technology Review named Antonio Regalado, MIT's Technology Review, had heard that something was going on concerning gene-edited babies in China. He's on top of this like no one else. So he went over to China and he found on a clinical trials registry this experiment going on. It wasn't the physician involved and his team did not want to disclose it, but Regalado was ready to break the news with the second international genome editing conference in summit in Hong Kong at the end of the month, and the AP had already been informed, and so the story came out with the beginning of the summit at the end of November, with Regalado essentially breaking the story. And since then, Regalado has tracked the number of scientific publications dealing with human embryos, and the current count is the U.S. 1, the UK 1, China 10. Why is all this happening? Well, I have a historical perspective of this, and as we related in our book, The Biologist's Imagination, China is wanting to restore its scientific glory of years past. It basically went insular in the first half of the 15th century, in the 1400s, and went insular and has been insular ever since, until now. And on that note, I will read a quick paragraph from our book, The Biologist's Imagination, which is about the history and current state of innovation in the biosciences, published by Oxford in 2014. It is a reminder that the scientific preeminence China once possessed, chronicled by the historian, biochemist, and embryologist Joseph Needham in his 7-volume Science and Civilization in China, has not been forgotten by China's scientific, business, and political elites. Quote, China has long been one of the richest, that is, one of the most fertile, best cultivated, most industrious, and most populous countries in the world, wrote Adam Smith in The Wealth of Nations. It seems, however, to have been long stationary, unquote. What was true in 1776 when Smith published his book, and for 2 more centuries, is true no longer. So this Thousand Talents program is beginning to take off. The Chinese government is putting a lot of emphasis on it. Here is Dr. He Zhonghui. I say that carefully because there are a lot of different pronunciations for his name, but I went to Chinese TV and listened in. So He Zhonghui. He is in his mid-30s. He studied at Rice, got his graduate degree there, a postdoc at Stanford. He is a biophysicist, not a doctor, not a clinician. He is an expert in genome sequencing, and he formed a company called Direct Genomics in Shenzhen, which happens to be, by the way, the place where the BGI, the largest sequencing factory in the world, is located. So not a coincidence there. In last August, Chinese state television was talking up this guy, and he went on TV and said, I did that. I built this sequencer. He built a sequencing machine that he claimed would outstrip all others available. Well, it turns out that the sequencing machine was a derivative machine from his Stanford advisor. So you get an idea that this guy wanted to go places, but he may be fast and not in the normal way. Okay, so he noted, and since this came out, that he was basing some of his decision in part on language in a National Academies of Science National Academies of Science criteria set forth in a major document that year about what was necessary to do before proceeding with clinical trials concerning human germline editing. So we're talking about basically the human germline. We're talking about eggs, sperm, and early embryos, not the body cells. So let me give you a quick rundown of a timeline that Anthony Regalado at Technology Review and his colleagues put together. November 25th, the bombshell. A team led by Dr. He at Southern University of Science and Technology in Shenzhen has been recruiting couples to create the first gene-edited babies. Specifically, they plan to remove the gene CCRS in hope of rendering the offspring resistant to HIV infection. Dr. Her claims to have altered embryos for 7 couples, resulting in 1 successful pregnancy and subsequent birth of twin girls. He posts videos to YouTube where he says the girls, who he calls Lulu and Nana, had been born 2 weeks earlier. The next day, November 26th, the disclosures provoke an international outcry. Many scientists within the field condemn Dr. He's actions, and Chinese authorities begin investigations of whether the experiment broke laws or regulations in China. In the US, one of the inventors of the CRISPR gene editing technique calls for a global moratorium on the use of the technology to create gene-edited babies. Rice University announces a full investigation into a professor who had assisted Dr. He with his research, actually his graduate advisor. November 28, He Jiankui defends his experiment at the International Human Genome Editing Summit in Hong Kong, saying, "I feel proud." He announces that a second gene-edited pregnancy is underway. November 29, Chinese authorities ordered Dr. He and his colleagues to immediately suspend their research activities, denouncing them as extremely abominable in nature and in violation of Chinese laws and science ethics. December 28, it is reported that Dr. He appears to be under house arrest in a university guest house in Shenzhen, guarded by a dozen unidentified men. January 18th, Chinese state media confirms the existence of the 2 gene-edited babies and says that Dr. He conducted the experiment for his own, quote, fame and fortune. January 29th, American Nobel Prize winner Craig Mello acknowledges he knew about the gene-edited babies while the pregnancy was underway. Mello had been a commercial advisor to one of Dr. He's companies, Direct Genomics. February 7th, Stanford University announces it has launched an investigation into what several high-profile faculty members knew about Dr. He's gene-editing work. February 20th, editors of the CRISPR journal unilaterally retract a paper on the ethics of gene editing published by Dr. He in November. The paper had made no mention of CRISPR babies. If he had followed his own guidelines, none of this would have occurred. February 21st, Dr. He's attempt to delete the CCR5 gene could inadvertently have changed the girls' brains in a way that affect cognition and memory. New research suggests, basically enhancing learning behavior. So essentially, Dr. He, that's how I'll refer to him, violated human subject research norms, informed consent protocols. He forged, so it is alleged, hospital ethics committee review documents that are necessary to do the IVF, the in vitro fertilization. And he skipped basic research and preclinical trials, which could have taken many, many years to do. He just kind of moved on. Why did he do what he did? Well, we don't know exactly. Not everyone was quick to condemn him. George Church, the visionary geneticist at Harvard Medical School, asked the question, is this a Jesse Gelsinger moment or a Louise Brown moment? Jesse Gelsinger was 18 when he was a subject of clinical trials. He had a genetic disease that affected his liver. He was otherwise healthy. He died as a result of the clinical trial. I think the use of virus to bring gene therapy to him backfired. Louise Brown. Louise Brown, of course, was the first test tube baby. Robert Edwards on the left, the physiologist with Dr. Patrick Steptoe, gynecologist, made it happen over 15, 20 years. Leslie Brown, Louise Brown's mother, and Louise's son, Cameron. This was taken in 2010. It's, I believe, the year that Robert Edwards won the Nobel Prize for Physiology or Medicine. All right, so he was Robert Edwards was a very interesting fellow. He was stricken by the plight of the infertile. Back in the 50s and 60s, the concern generally was not infertility. The concern was overpopulation. You remember Paul Ehrlich's The Population Bomb, 1968. That was the concern. Basically, people who were infertile were making a contribution. They didn't even realize it by keeping the population down. Well, that didn't please them particularly. So he did a lot of preclinical work over many years, basic research and preclinical work. In particular, what they call capacitating the sperm. You can't just put sperm in a petri dish with egg and expect fertilization. The sperm have to be primed essentially with uterine factors to be activated. And he figured it all out. And his colleague Patrick Steptoe, as the gynecologist, was a leader in laparoscopic extraction of eggs. 200. All this was done with informed consent of the as was practiced at the hospitals at the time. 282 patients, 457 laparoscopic cycles. Finally, 2 healthy babies. The first being Louise Brown in 1978, reported immediately in the journal Lancet. The first 2 of more than 8 million IVF individuals now. Louise Brown is 40 years old. What about when she's 50? She and other IVF, are they going to be susceptible to anything in particular? They're IVF. This is an experiment. We don't know. There's no reason to think there would be any more than anyone else, but we just don't know. So it is, in a sense, an ongoing experiment. The Medical Research Council in the UK wouldn't fund this work. The professional medical societies kept their distance from it. There were all sorts of claims that this was going to lead to terrible things. The news media was all over it, as you can imagine. Dr. Edwards did not believe in going underground. He believed in working with the news media to try to explain what they were doing. James Watson, of DNA double helix fame, wrote in the Atlantic, "This is going to lead to human clones." He condemned this work in Congress, saying that all sorts of terrible things are going to happen here in the U.S. and around the world. A leading developmental biologist was certain that monsters would be created. The New York Times talked about brave new babies and on and on. So that was the context for when Louise was born, and when she was born, things flipped very quickly. She was a healthy child, an infant child, has been ever since. Basically, all of that great overwrought fear and concern didn't entirely disappear, but it subsided dramatically. All right, now let's go fast forward to the 1990s, and we have the first test tube baby, in vitro fertilization, Louise Brown, and we have here Adam Nash, that little infant there in John Wagner's arms. Adam Nash is the first savior sibling. So Molly Nash on the left, a child with Fanconi anemia, born in 1994. This photo in 2000. She was not doing well. Her bone marrow was failing, which is typical of young kids with this disorder, Fanconi anemia, risk for pre-leukemia, leukemia, lots of other problems. And Wagner was determined, John Wagner of our pediatrics department here at the University of Minnesota, and also the stem cell, umbilical cord stem cell authority, was approached by Molly's parents because of the reputation that we have in bone marrow transplantation and in blood-forming stem cell transplantation. We pioneered bone marrow transplantation in 1968 under Robert Good. We have a strong program here. They came here, and he imagined a way to deal with this situation. Basically, having Molly's mother create embryos that could be then tested through a process called pre-implantation genetic diagnosis. It would be quite interesting. It was just available at that time, not before, where the embryo at the 8-cell stage called a morula, you could extract one cell out of the 8, test it for genetic disease, or in this case, pioneering tissue match. They did that, and Adam was a perfect match in terms of HLA tissue, and he did not have the Fanconi mutations. So they proceeded. the pregnancy was a very difficult and testy pregnancy for Molly's mother. But in the end, there's Adam, and in a month or two, he is providing umbilical cord stem cells to rebuild his sister's bone marrow and blood system. Molly is now 24 and in basically good health, although she's got Fanconi anemia, and that comes with other things besides risk of bone marrow failure and leukemia. And her little brother Adam is 18, and unlike the concern expressed by many bioethicists back at the time about this, the emotional dimension of a brother giving a sister life, essentially, they get along quite well. There has not been any, psychological dimension of concern to bioethicists back at the time that this happened. But I could tell you that that this turned over the whole bioethics community. What do we do? Parents want to save their child. I mean, yes, absolutely. However, how far should we go as a society, etc.? All right. CRISPR. Who's heard of CRISPR? No, everybody, I hope. Okay. CRISPR is probably the leading. It is the leading genome editing technology. It comes from bacteria. It is a bacterial adaptive immune system. It is a remarkable system for protecting bacteria against viral attack. And basically, the system is such that a snippet of viral DNA is captured in the chromosome of a bacterium. That is the memory for future attacks. And when a future virus comes along, and there's a match to that particular sequence, the bacteria can go into action, send an enzyme called Cas9 to the specific target site in the viral DNA, attack, chop it up, virus is gone. In 2012, Jennifer Doudna and Emmanuel Charpentier, Doudna at UC Berkeley and Charpentier then at the University of Vienna, if I recall, basically devised a way to sort of commandeer this system to enable the Cas9 enzyme, the DNA cutting enzyme, to be put anywhere in any DNA sequence of any living thing. Okay, so they were working with bacteria, and then soon others, Feng Zhan at Broad Institute, working with mammalian cells. It showed this is a universal approach. Program a sequence, as to say, develop a sequence. It's called a guide RNA sequence that's programmed to go to any place in the genome that has complementary letters. Attach the Cas9 to that guide RNA, and so it becomes a complex. And then the guide RNA will take that Cas9 cutting enzyme to any place in the genome, and go into action, create a double-strand break, which the cell then has to repair. Now, this talk is about germline editing, but it applies to germline editing too. That is to say, sperm and eggs and their precursors, spermatogonia, oogonia, and early embryos. It's the same process as would be with our somatic cells, with our body cells. All right. This is a way, in fact, of steering our genetic destiny. Once the cut is made at a specific sequence in the DNA is a double-strand break, so it's cutting both strands of the double helix. The cell has to repair itself, otherwise it dies. There has to be repair that steps in, and that's where the editing really comes into play. Okay, let's talk a bit about why would anyone want to edit embryos as opposed to creating embryos and then doing the PGD, the pre-implantation genetic diagnosis. You just test this embryo. Francis Collins at the National Institutes of Health believed this is the way to do it. just select the embryo without the genetic mutation or the disease, and that's a tissue match, and go forward from there. Others, George Daly, the dean of Harvard Medical School at Stem Cell Pioneers, says no, I think we have to develop translational guidelines for clinical trials. But this is important to do because PGD doesn't work in every case. There is the issue of being able potentially to remove the disease from the entire human gene pool by working through germline, because after all, changes made in the germline are carried on through future generations, unlike changes made in let's say, our liver cells or other tissues in our body, which leave with us, essentially. So it's the idea of altering the germline, but could it be done in ways that essentially remove awful diseases, some of which are compatible with life only for a few months, from the entire gene pool, plus the opportunity for affected individuals to not only be treated, so essentially as embryos for their genetic disease, but also to be able to then become parents themselves and not worry about passing passing on the disease. All right, so we're talking about 6,000 or 7,000 genetic diseases here. Diseases like sickle cell anemia, cystic fibrosis, point mutation in both cases, one letter. Just change one letter in the right cell type, of course. Duchenne muscular dystrophy, Tay-Sachs disease. The risks are off-target effects, so that the guide RNA doesn't go just to where you want it, but it may go off yonder and in another part of the genome and make a change that you don't want. So that's one risk that has to be guarded against. A second is mosaicism. So if you edit the embryo at the 2-cell stage, let's say, and one cell is edited and the other is not, then eventually you're going to have what's called mosaicism. You're going to have an organism that is partially edited, depending on the tissue and the place, etc. But that is not a ideal therapeutic approach in that sense. The immune response to CRISPR or other genome editing technologies is just beginning to be understood now. So there is an immune response. Is it manageable? Well, the advocates in the field certainly think it is a manageable problem to deal with. Okay, but I'm going to make this point, and I hope it registers. CRISPR, we're in the CRISPR revolution. I've been working in the medical school since 1983, and I've seen a lot, and some amazing things happen. And maybe I'll talk about them briefly just ahead. But without DNA repair, you don't have anything, because the cell dies. The cell has to be allowed to repair itself. So I've got a 2-minute, 3-minute video here that helps you understand. And this comes back, this is very important, this comes back to Molly Nash's disease, Fanconi anemia.
CRISPR-Cas9 genome editing in human cells occurs via the Fanconi anemia pathway. The genome editing tool CRISPR-Cas9 has the potential to treat a range CRISPR-Cas9 genome editing in human cells occurs via the Fanconi anemia pathway. The genome editing tool CRISPR-Cas9 has the potential to treat a range of genetic diseases. Scientists have learned how Cas9 locates and cuts a specific sequence of DNA, but that's only half the equation. Once the cut is made, we need to understand how the DNA break is repaired in order to effectively add, subtract, or change the genetic letters. Now, Jacob Korn's lab at UC Berkeley shines a light into this black box and reveals how a little-known DNA repair pathway could play a big role in the success of genome editing therapeutics. After Cas9 makes a cut in the cell's DNA, the cell can repair the break in a variety of ways. One repair method involves patching in double-stranded DNA that's been added by the experimenter. This DNA is designed to have long ends that match the sequences at the cut site, telling the cell where it needs to go. A different pathway, called single-strand template repair, or SSTR, allows a cell to insert single-stranded DNA with shorter matching ends. Experiments show that cells can insert single-stranded DNA at cut sites more efficiently than double-stranded DNA. But how? To answer these questions, a scientist in the Corn Lab named Chris Richardson turned off specific genes across the human genome to see if any of them were essential for single-strand insertion. Richardson observed that when he turned off genes involved in the Fanconi anemia pathway, the cell was unable to repair the Cas9 break using single-stranded DNA. Further experimentation revealed that FA proteins migrate to the site of the Cas9 break and act like a traffic signal to direct repair pathways like SSTR. Understanding the role of the FA pathway in repairing Cas9 breaks gives scientists an upper hand in developing genome editing therapies. By controlling the traffic signal, scientists may be able to direct DNA repair towards the insertion of healthy DNA with SSTR, instead of random mutation, a choice that is currently difficult to control but essential for one day curing genetic diseases. The black box of DNA repair mechanisms just got a bit brighter.
Okay, I want to mention that the Innovative Genomics Institute is where Jennifer Doudna, the leading figure at UC Berkeley in the whole CRISPR revolution. That is her institute. So these people are being funded by her institute there in part, and this is leading work. And the important thing to remember here is, again, no DNA repair. There's no CRISPR babies in the news, no genome editing for treatments and cures, no food crop improvements, no infectious disease and invasive species control through this new technology, all of which are in play, provided we understand how DNA is repaired and can exert some control over that. And we're going to learn more about that now, especially the Fanconi anemia pathway, because that's a key. And I think that and other, actually, Dr. Richardson, who did this work, believes that our own Fanconi anemia pathway, just named that because of the disease Fanconi anemia is affected, mutations affect that pathway. That our own pathways may have a lot to say about how successful these therapies become, because that's going to be key. It's possible to imagine that we could actually, through genetic expressions advances, improve our own pathways, and that would enable these genome editing therapeutics to really move forward in a remarkable way, I think. And it's also important to remember that Lulu and Nana, without their DNA repair, without the ability of that mutation that they wanted to insert in the CCR5, actually Delta 32 mutations called there are some 10% of Northern Europeans have this mutation. If that editing is successful, we won't know. We may never know, by the way, about Lulu and Nana, although it's going to be hard to keep it a secret because, as the National Choir says, inquiring minds want to know. So it'll be difficult to keep it a secret forever. But if it's successful, it'll be because the DNA repair incorporated the mutation and prevented the proper functioning of the CCR5 gene, which then prevents or is resistant to HIV infection. All right. Actually, I have a little bit of a place here too. Don't worry about this. I'm not going to go into it. But this is the human genome. Okay. And these other chromosomes, it's called an ideogram. Normally, the chromosomes come in the diploid, come in pairs, right? We have one from each parent in our cell nuclei. And I worked with the leading authority, really, at the time, high-resolution chromosome banding back in the 80s. And so all the genes are there except for 37 that are in the mitochondria that are floating out in the cytoplasm, the little energy factories. All the CRISPR targets for genetic therapies are therein. Now, I was trying to think, how can I convey distances here? All right, so if you're, let's say you're at 5,000 feet looking down in your house, okay? So you're at 5,000 feet, now we're at 5,000 feet. And if you want to find CRISPR, you have to go to your toaster. You have to zoom into the toaster, then from 5,000 feet. All right, that may be a little bit exaggerated. The point is that's molecular. This is chromosomal. But you can learn a lot from chromosomes because chromosomes break and rearrange in different types of cancer. Fanconi anemia is one of several what are called chromosome breakage syndromes. And I was working with Dr. Yunus, not specifically on Fanconi anemia, but on chromosome breakage generally. And chromosomes do break and rearrange in very predictable places in different kinds of leukemias and lymphomas. He found in normal cells that he put them under stress by depriving the cells of the ability to replicate certain enzymes. If you use radiation on the cells, if you deprive the cells of folic acid, which is a B vitamin and critical for DNA synthesis and repair, if you put the chromosomes, normal chromosomes, through a stress test, you'll find that they break, little gaps and little breaks in different parts of the chromosomes, and these are the hotspots, and basically what chromosomes break and rearrange in different types of cancer. So he found that test in fragile sites was important to understand what's called genomic stability, and Fanconi anemia is a genomic instability syndrome because there's a problem of repairing any number of different types of breaks, but in one in particular called an interstrand cross link. So think of yourself as zipping up your jacket, and you come to unzip it, and your scarf threads get caught in there. If you want to unzip the jacket, it's caught, gets hung up. And this happens to me all the time. Mostly when I'm zipping up, but even when I'm zipping down. All right, so there's that little spot there. It's a snag. Well, in DNA, you have a bond between a bonding that takes place that is not normal, but there it is. And unless the Fanconi anemia team can show up on site and repair the job, you're going to have problems with the chromosomal structure and its integrity. It's going to break. So that's in Fanconi anemia. That's the key finding. But it also, as you saw in the animation, Fanconi anemia pathway will come in and repair different types of breaks. And some of these breaks are very critical for developing, understanding them and how to resolve them. Because one type of break can be repaired, what's called non-homologous end joining. Basically, the two ends come together. Maybe some DNA is lost, maybe some is gained in there, but there's no rhyme or reason. It's just reunited. However, if you can do therapeutics, you want to have a specific sequence that goes in there that may repair a cystic fibrosis point mutation, or a sickle cell point mutation, or a Huntington's disease of 3 or 4 letters. You want to have to replace the sequence, make it homologous, the same, but you want to be able to have that incorporated. And that's where the Fanconi anemia pathway is marvelous, because it allows you to create, you can send in an oligonucleotide, basically a synthesized piece of DNA that you carefully constructed. You can send that in, and the Fanconi anemia pathway in repair will take that strand and put it right where it belongs, repair, and then you've got a cell that will divide. As long as you're doing it in the right tissue at a high enough percentage, you can imagine genetic repair of complicated diseases. I'm coming to the end. I'm going to move through quickly now because
We'd like to leave some time for questions.
Okay.
So
Why don't we? Well, let me move through a couple more slides, and I won't talk about it. I'll just show you. I'll pass through this one. Basically, it compares computer performance improvement in RAM with DNA breaks over 40 years or so. And it's important to remember that with stem cells, it's possible to imagine gametes, that is, say, eggs and sperm, being created from your own body cell, from skin cells. And Hank Greeley at Stanford University, a bioethicist and professor of law, thinks that eventually, in 20 or 40 years, that's how babies will be created. That is to say, through not in the laboratory and not by conventional means. And the human embryo is off limits to U.S.-funded researchers. That has been the case since 1996. A map that I created showing comparative ethics and guidelines from around the world. This was used in the great stem cell debate in 2007 in the U.S. Senate, showing basically what's allowed and what's not allowed in each country in terms of stem cell, embryonic stem cells. What you could do, what you couldn't do. At that time, we were fairly restricted. It was during the Bush administration. A couple dozen stem cell lines were used. I bring this up because the whole issue of human germline editing has been now brought up with WHO, the World Health Organization, and they are not going to be looking at the middle of this month. They've impaneled a committee of 20 or so people, co-chaired by Margaret Hamburg, former FDA commissioner in the Obama administration, currently the president of the AAAS, American Academy for the Advancement of Science. She's going to co-chair this. I sent this map to her last week, and she responded. She wanted to know if this, if I'm still maintaining it. All right. Huxley, you can't get away from it in this brave new world. I won't talk about it, but just do reread the book I just did. It's amazing to see. After all, it begins in a London hatchery, a London embryo hatchery, a mass-produced world. And that, I read it years ago, and I've almost forgotten now. It's kind of opened my eyes. All right. And then lastly, of course, Time Magazine. If you have questions about the uterus transplants and the three biological parents, I'd be happy to answer them. But I'll stop there. But with one last comment that I decided I'd throw at you. In what moral universe is it okay to alter the genetic destiny of future generations? Well, we've been doing just that to other living things for a long time, altering their genetic destiny. With germline editing, we now have become the first species on Earth, so far anyway, to read, write, and edit its own genetic destiny letter by letter. So thank you very much, and please, any questions?
We have time for a few questions.
Yes.
On the Chinese gene splicing experiment, what were they trying to address with that splicing? Was it a specific thing they were going after?
Well, the CCR5 gene, which is on the CD4, a specific subset of T cells, lymphocytes, that HIV/AIDS patients back when we didn't have treatments, that cell was destroyed. The virus would enter that cell through the membrane and destroy that particular cell, and then the entire immune system would crash. Okay, so the idea here is to disable the CCR5 gene. It's naturally disabled in 10% of Northern Europeans, as I mentioned, and disable that, and those patients then become resistant to HIV infection, which is a major stigma in China, as it is other places, but it's a stigma there. That's the ostensible reason. But Dr. He basically carried forward an experiment that was really medically unnecessary. That's important. That's one of the important things. The technical brilliance kind of thing got the best of him, I think, and all the accolades that come with technical brilliance. The fact is, there are other ways to deal with HIV risk, sperm washing, what have you. This was basically an effort to showcase his talent.
It's a noble cause.
Noble cause. I mean, it would be good to learn how to disable the the CCR5 gene. They are working on that, using CRISPR to work on that. But to do it in a patient as an experiment is,
Just on that,
Yeah.
on that experiment, maybe inside the ethics or how is he going to determine if he's successful? Because you won't.
this is a fantastic question. The government, of course, has now got these girls in its custody. It's managing the situation. There can be no disclosure. As far as I know, maybe Antonio Regalado has better information, whether they have assigned physician scientists to follow these. It's not going to be published, so no one's going to know. Well, I mean, unless there are some enterprising reporters in China.
In the Chinese twins now, at what point was this done? At fertilization or first division or what?
It was done at the 2-cell stage, as I understand it. There were 2 embryos. Well, I shouldn't say that. I'm not positive, but it was at the very early. So right after fertilization, it was done. And then PGD, pre-implantation genetic diagnosis, was done, according to Dr. He. But none of this has been published. She just said that at the meeting, that he did PGD, and he sequenced full genomic sequence of pulling out one cell and doing sequence. Perhaps it's not the 2-cell stage, but early on, what he found is that everything looks okay with the one embryo. The other embryo, there is what's called mosaicism, which means that the edit didn't take in all the cells of the several cells that I think it's called a blastocyst, which is an early embryo.
Go back to a big question. 40 years ago or so, Christian Bernard with the heart, the technique, the surgical technique wasn't in question. The question was organ injection. That had not been perfected yet. And he went ahead, did the process.
Yeah.
The patients died,
Yeah.
and his rationale was, well, of course, the issue.
Yeah.
And somebody had to step forward and say, we're going to sacrifice some people on the force of the issue.
Yeah.
Is this what we're dealing with now?
Well, I mean, I'm assuming that that patient gave informed consent. And now that's always an issue, because what happens is people give informed consent, and then after the fact they decided, well, they didn't really, and that's why informed consent has to be done with not just with Dr. He and his team, but I mean, other people, should be brought in to ensure that that this is being done appropriately, because there is a way. If you're a scientist, you're going to want to do the experiment.
But are we saying in this talk that this is going to be done someplace, somehow? The ethical question is just going to be pushed aside.
I would say that based on my experience with that stem cell map and the international effort to get everybody on board with a single way of moving forward, of course, that did not happen. The UN did propose a human reproductive cloning ban, and that was signed on to by many countries. That was an easier call than banning or restricting human embryonic stem cell research. We didn't want to. Nobody really, no sensible scientist wants to create cloned human beings. Although there was talk back then that somebody had done that. Of course, it was not true. The other thing is that if you step outside of the scientific norms, you run risks. And I gave a talk at Stem Cells Asia in 2010 in Seoul, based on this map work. And this was right after the Great Korean Cloning Scandal. Wang Woo-suk, a veterinarian, claimed that he had cloned human embryonic stem cells. It turned out not to be true. He had gotten eggs from his technicians, to do the supposed cloning. He did clone dogs, but he didn't clone human embryonic stem cells, as it turned out. And then this led to a major scandal. The Korean government, I remember, had a postage stamp of a fellow getting up out of his wheelchair and step by step, walking. As a result of this wonderful advance in cloning. So the government had gotten behind this man, and it turned out to be a disaster. What happened to him here? Well, he stripped of a lot of his positions in the scientific community in Korea. But of course, China, they thought he was doing great work, so they brought him over there, and when I proceeded with his cloning experiments.
Last
Are
question.
these same techniques of gene editing that we're talking about as an advance in humans, is that already being used in animals such as cows?
Yes. Yes. In fact, we at the University of Minnesota, we have tremendous science in this area. CRISPR, yes, but also a earlier technique called talons, and we're really good at that. And our people, particularly Perry Hackett, who is at the College of Biological Sciences and one of the founders of the Genome Writers Guild, has provided technologies that allow the not only for these gene editing tools to be put into the cell by novel means rather than viruses, but also Perry has developed the zebrafish animal model. And I just saw a paper where the zebrafish served to elucidate the Fanconi anemia pathway because it's almost the same as ours. And so these are rapidly reproducing creatures who can serve genetic testing very well. Plus, we have advances in food crop plants and livestock, hornless cattle, for example, so you don't have to cut off the horns, or soybeans that have certain advantages over conventional soybeans in nutritionally. These things are being done, and the University of Minnesota, in the ag area and livestock area, is really quite far along. And this Genome Writers Guild is a consortium of the University of Minnesota, the Mayo Clinic in Health, Iowa State and Ag, and so on. So we're right out in front in some of these areas.
Is this directed at conception or around that period? Is there a way that people can be treated later using gene editing?
Yes. My talk was about germline editing, but the answer is absolutely. And in fact, the germline editing is just a high profile dimension of a much larger possibility, which is somatic cell, which is body cell editing for any number of conditions, autoimmune conditions, certainly cancer, neurological conditions. Basically, you have now the ability to go any place in the 3 billion base pair human genome and make changes, and you can do it fairly reliably. But you're not going to succeed unless you understand the DNA repair and how it's done and how proper and correct it can be made.
Well, I would say we have a lot of information to think about on this Monday morning. Thank you so much. Can you stay around for a few minutes afterwards?
Sure, of course.
If people have questions or want to talk to Bill, please feel free. Thank you. Thank you. Oh, that was fabulous. Thank you so
Was it okay?
much.
I didn't get too far off. I went over the